Welcome back to another presentation here from Behavioral Health 2000. Today we deal with Alzheimer's disease. It fits into a larger context of unresolved and untreatable or ill-treated psychiatric and brain disorders such as depression, schizophrenia, addiction, and autism. And you see some of the compilation of the numbers here in the first slide. Alzheimer's disease is estimated to reach 115 million people by the year 2050 and is almost certain to bankrupt our healthcare system. I will show you a brief video that is produced by the Alzheimer's Association that gives you their thinking and some contextual and emotional background as to the dramatic effects that this illness has on those affected, of course, and on caregivers and family alike.
One disease, millions of lives permanently disrupted. Today, there are over 5 million Americans living with Alzheimer's disease. But Alzheimer's is taking more than memories. It's taking lives. What few realize is that Alzheimer's kills more people than breast cancer and prostate cancer combined. And Alzheimer's is the only disease in the top 10 causes of death that can't be slowed, stopped, or prevented. From 2000 to 2013, while deaths from other diseases decreased significantly, Alzheimer's deaths increased 71%. But equally disturbing, many living with Alzheimer's say they are not being told their diagnosis. While more than 90% of people with the most common types of cancer say they are told their diagnosis, only 45% of people with Alzheimer's or their caregivers say they are told. The economic toll is staggering too. In 2015, it will cost the nation $226 billion to care for people with Alzheimer's. By 2030, 72 million baby boomers will reach an age of greater risk. The fact is, everyone's at risk for Alzheimer's, even you. But everyone can help, especially you. We're the Alzheimer's Association, the brains behind saving yours. Join us.
So welcome back from the video. In this slide you see the projected prevalence and cost of Alzheimer's disease. An exponential increase, reaching 40 million people by 2060, and a healthcare cost in the trillions of dollars that we certainly will not be able to pay. So a lot of money and effort has been expended on the research in Alzheimer's disease. What is going wrong? And I will now show you a brief video that we prepared on a different occasion, namely the amyloid hypothesis of Alzheimer's disease. And in this video, we give you some clue as to what might be amiss in this hypothesis, which has directed so much research and so many clinical trials in an attempt to remove amyloid plaques from the brain of Alzheimer's patients. Welcome back. Today we have another installment on our ongoing series on Alzheimer's disease. In particular, I like to focus on the notion that perhaps we need to reject the well-established amyloid cascade hypothesis of Alzheimer's disease. As you know, we have done 2 previous presentations on breakthroughs in Alzheimer's disease, describing how a monoclonal antibody was able to remove a great deal of the amyloid deposits in Alzheimer's patients. And in another presentation, a small molecule was discussed, which is an inhibitor of a protease, which also is able to clear amyloid deposits from Alzheimer's brain, all of which seems to support the notion that the amyloid hypothesis of Alzheimer's disease should reign supreme, and it really has elicited all the funding in the world And many drug companies have literally poured billions of dollars into researching that particular aspects of Alzheimer's disease. Now today, I will try to undermine in a progressive way the notion that the amyloid cascade is the only story, or perhaps even not the determining story in the pathophysiology of Alzheimer's disease. And towards the end of the talk, we will arrive at a paper which was just recently published from a lab at Stanford, which sheds entirely new light on what might be going on not only in Alzheimer's disease, but in a number of neurodegenerative disorders, such as amyotrophic lateral sclerosis and Parkinson's disease, in addition to Alzheimer's disease. There may perhaps be a common pathway in which all lesions that eventually lead to neurodegeneration end in the activation of what is called M1 astroglia, neurotoxic astroglia. And we will approach this in a stepwise fashion. But first, let's talk about the amyloid cascade hypothesis of Alzheimer's disease. So the way it's classically stated is that over time, an imbalance in amyloid beta production and or clearance leads to gradual accumulation and aggregation of this peptide in the brain, then initiating a neurodegenerative cascade that involves amyloid deposition, inflammation, oxidative stress, neuronal injury, and so forth. So the amyloid hypothesis for Alzheimer's disease posits a neuron-centric linear cascade initiated by A-beta deposition and leading progressively then to tau pathology, synaptic dysfunction, inflammation, neuronal loss, and eventually dementia. Now, if this is all true, then why is there no good correlation between the levels of amyloid beta in the brain and cognitive impairment? In fact, the correlation is quite poor. The evidence overall seems to suggest that there is a long, complex cellular phase consisting of feedback and feed-forward responses involving astrocytes, microglia, and the vasculature as well. So the amyloid hypothesis has no explanation for the silent incubation period of Alzheimer's disease. Even in those cases of familial inherited Alzheimer's disease with early onset, why is it that you have the genetic lesion from the womb and childhood? Why is it that it takes decades to develop the pathology if the genetic lesion alone is the driving cause? So although abnormal amyloid beta production is already in place from conception, the toxicity manifests itself only in midlife. So there's no good explanation that can be derived from the amyloid cascade hypothesis alone. Okay then, so here is the key element, the amyloid precursor protein, which is a protein in the membrane of the cell incorporated into the membrane here. You can see there are various points where cuts can be made and certain fragments of this huge protein can be released, in particular the so-called AB42, which is thought to be the main culprit in leading to aggregation and the precipitation of plugs that then poison the neuron, leading to downstream consequences that we will discuss shortly. We have genetic forms of Alzheimer's disease that are inherited, and these affect the enzymes that are involved in the cleavage, in the release of these amyloid peptides from the membrane, namely presenilin 1 and 2 genes. And if those are incorrectly encoded, we can increase A-beta 42 production throughout the lifetime, which leads to oligomerization, precipitation, and amyloid plaque formation. But the vast majority of cases of Alzheimer's disease, of course, are so-called sporadic. There is no family history or genetic background. And here there must be other factors in place. For example, a lack of clearance, where the normal turnover of these peptides is gone awry, for example, by the inheritance of the ApoE form gene that has been implicated in mediating an erroneous removal, or deficient removal of the A-beta peptide from the brain. So here then is yet another summary. So we have the subtle effect first of A-beta oligomers on synaptic activity, on the firing and processing of nerve impulses. Gradually we get deposition of the A-beta 42, which in fact can activate microglia and cause an inflammatory response, which can have secondary destructive influence on nerve cells. Eventually we wind up with a change in kinase and phosphatase activity affecting the molecule called tau, a molecule associated with important functions in the brain that can be phosphorylated, and when hyperphosphorylated can precipitate and form the so-called tangles. And then this synaptic destruction then leads to the occurrence of dementia down the road. In the next slide, I introduce you to the gene, the amyloid precursor protein gene, about which we will say more soon in this lecture. You can see it's located on chromosome 21. It has a large number of introns and exons. You know that genes are organized according to coding regions and non-coding regions. The non-coding regions are being spliced out at the level of messenger RNA. You can see in the bottom part of the slide that there are different splice variants. So there is already a hint that the gene can produce different forms of the amyloid precursor protein, labeled here 695, 751, and 770, depending on how the splicing of these different introns and exon segments is accomplished. In the next slide, you see the timeline that we need to, that any theory needs to explain. We can detect the AB42 in the spinal fluid here on the left-hand side early in the game, decades before dementia symptoms occur. By PET scanning, we can detect the amyloid plaques in the brain, likewise decades before symptoms occur. Cognitive impairment occurs here. Now, if you have high risk, for example, because of the inheritance of the ApoE4 gene, your onset of Alzheimer's symptoms will occur much earlier, whereas if you are endowed with low risk, it will occur much later. But eventually, we all may be at substantial risk of developing Alzheimer's pathology because of aging. Our population is getting older, and age is the major risk factor for Alzheimer's disease. So any amyloid theory needs to have an explanation for the age dependency. Now we will now go to the story at hand here, a new revised amyloid story that may explain more of what the observed data show and fill in some blanks. It is also quite revolutionary because it topples several dogmas and preconceived notions that we had about genes in the brain. So here's the gentleman who put this on the map. It just appeared very recently, a couple of weeks back, towards the end of the year 2018, in Nature, as a Nature article. His name is Gerald Chung, and his group members are listed here. He is down in San Diego, and he has discovered that the amyloid precursor gene can undergo recombination in nerve cells. In other words, fragments of the gene can be recombined in different patterns. Now, this recombination of genes is very common in cells that are involved in creating a new organism. When you combine eggs and sperm, you have an event where the cells divide, and this replication of DNA, which occurs in the context of this, frequently leads to recombination and reshuffling of gene fragments in different ways. Now, it's not supposed to happen in mature cells, especially in nerve cells, which don't divide. They just sit there and work for their entire lifetime. Now, how did Dr. Chung come up with this? I will now show you a brief video that has him explain the work and the approach that he took and the ideas that led him to this remarkable finding.
Our research provides hope that we have a near-term medicine to treat Alzheimer's disease. My name is Gerald Chung. I'm a professor and senior vice president for neuroscience drug discovery here at Sanford Burnham Prebys Medical Discovery Institute. Alzheimer's disease is the most common cause of dementia in the US, affecting some 5.7 million people, and that number is going to be going up very rapidly in the coming decades. Alzheimer's disease is devastating to the individual. It falls into two basic categories: those that are inherited and those that are not. The inherited forms account for perhaps just 1%. That raises the question of what's going on in that other 99% of cases. What one has been taught in school for many, many years is that every single cell in your body has an identical blueprint, identical genome that started from that initial fertilization step. What we found is that they change the very blueprint of a given cell. That is one of the core implications of the study that we've just reported. Scientists have referred to this as gene recombination. Gene recombination is a natural process, and in fact, it is the basis of our immune system. Gene recombination is a process where a stretch of DNA is cut, and then another piece can be brought to that cut piece to form a new DNA sequence. This is very similar to bringing together pieces of film. You can splice it together, and that would form your new movie or your new edited movie. The key finding from this work is that gene recombination occurs within the brain. It involves this gene referred to as APP, or amyloid precursor protein, that then can form the amyloid plaques, thought to be a major cause of Alzheimer's disease. Alzheimer's is a disease where recombination goes awry. Identifying gene recombination also led to an understanding of some of the very basic mechanisms that produce the recombination. There's an enzyme that we identified that is well known to many biologists, called reverse transcriptase. And this enzyme is quite famous because it was the first to overturn the so-called central dogma, going from DNA to RNA to protein. The reverse transcriptase will take an RNA and turn it into a DNA, so it goes backwards. And why is that interesting? Well, it suggests that you might be able to interrupt that enzyme's activity and then actually bring about some therapeutic advantages. HIV is treated in part by inhibitors of this reverse transcriptase. We then proceeded to look through the literature at individuals that were in that 65-ish range or older, of which there are approximately 100,000 taking these reverse transcriptase therapies for many years. We would have predicted there should have been 2,000 to 3,000 people with Alzheimer's disease in that age group. And what we found in the literature was at this point there had only been one report of Alzheimer's disease in that HIV population. The exciting possibility is that we could take already FDA-approved agents that have been in humans in some cases for decades and test them in actual Alzheimer's disease patients. It would be remarkable if it turns out that HIV, this terrible disease, gives rise to the first effective treatments for Alzheimer's disease.
Okay, welcome back from Dr. Chung's video, and we will now throw another curveball into this, namely the idea that HIV drugs that inhibit the retrotranscription of RNA into DNA that the AIDS virus uses to introduce itself into the human genome, that those drugs may in fact have utility in Alzheimer's disease. Now, these are drugs already FDA approved. They are on the shelf. We could use them now. So we need to explain why Dr. Chung believes that this might be something of utility and the ideas behind this. In the next slide, I show you kind of the key finding. He has found, in looking at postmortem Alzheimer's brain, that 100% of the Alzheimer's disease brain samples obtained an overabundance of distinct APB gene variants compared to samples from normal controls. And when the group sequenced all these different variants, they sure enough discovered all the single nucleotide changes identical to known mutations that have been previously described in familial Alzheimer's disease. The difference being that this occurred in a mosaic pattern. In other words, only some nerve cells showed this occurrence of reshuffling and recombination, but others did not. So whereas in inherited disease, all cells carry the defective gene, here a gene deficit or a defective gene is introduced in some way in only some cells, but not in others in the brain. This gentleman here, David Baltimore, in the 1970s discovered that viruses, certain viruses, can do something extraordinary. And he turned over the so-called central dogma of molecular biology, which I'll show you. And the central dogma went like this, first formulated in the 1950s. DNA, which contains your genetic information, is transcribed into RNA, which upon export into the cytoplasm of the cell engages the machinery that uses the information to produce proteins. So he discovered then that there are occasions upon exposure of cells to certain viruses, namely retroviruses, that RNA could be retranscribed, retranslated into DNA, which then could be inserted into the host cell and give the virus a permanent home in the genome of the infected cell. And here is a page from the notebook of Dr. Baltimore. What he did was he set up an assay where he loaded a precursor of one of the bases that is required to make DNA. And you can see on the left-hand side, he added all the different components, and he showed that there must be an enzyme. At the bottom you see his comments. There is an enzyme, he says, that is able to take RNA and write DNA from it. So that was the essential discovery. And this is what will now play a role in the work of Dr. Chung. In the next slide, a further explanation on mosaics. We have encountered mosaics before in the brain, For example, we gave a talk on mosaics in the brain a while back. I will show you a brief video excerpt from that. This is the group of Fred Gage at the Salk Institute. They have discovered that the so-called line repetitive elements can be inserted into certain nerve cells, not in others, in a mosaic kind of a fashion. And this apparently is dependent on the quality of maternal care that the rat pups had received. So there is a talk on this material that we prepared earlier. Here then is the story that Fred Gage's group was dealing with. He believes that maternal care can alter genomic structure, and the mechanism of this will be to activate latent transposons, especially of the L1 type, because the methylation of certain receptors and promoter sites that promote the production of these transposons might be deficient in rats exposed to poor mothering. And this is exactly what would then lead to the phenomenon of mosaic cells, namely cells that have certain genes expressed, neighboring cells, different genes expressed, both resulting from the same progenitor cell. So the progenitor cell is then modified by the random transposition of these line 1 repetitive ancient elements into new DNA targets, perhaps enhancing or minimizing or even destroying the activity of certain genes. Okay, welcome back. Now, the paper that we are discussing today had a precursor paper. Namely, Dr. Chung and his group looked at the amount, the copy number of the amyloid precursor protein gene in individual nerve cells in postmortem Alzheimer's brain. And what they discovered was that there are nerve cells which have an excessive number of copies. You should have only one copy from your dad and one copy from your mom. That's it. That is a normal state of affairs. But apparently there is a mechanism in place in Alzheimer's brain which allows for many copies of the APP precursor gene to be somehow inserted into selected cells of the brain. Now that is a disturbing finding, because we know that, for example, in trisomy 21, where you have an extra copy of the APP gene in Down syndrome, that Down syndrome predisposes to Alzheimer's disease with a very high penetrance and frequency as Down's patients become older. And they are becoming older these days because care has improved and they live a longer lifespan and frequently come down with dementia of the Alzheimer's type later in life, attributed to the extra copy of the APP gene that they inherited with this extra chromosome 21. So here then, in this case, in the postmortem Alzheimer's brain, there are no extra chromosomes, but something is in place, some mechanism is operating that apparently is inserting extra copies of this potentially deleterious gene into some nerve cells. So this then was the starting point for Dr. Chung. And here then is the paper which appeared in Nature. So now we understand what he's talking about. Somatic, namely not germ cells but mature neurons, somatic amyloid precursor protein gene recombination in Alzheimer's disease and normal neurons. That's what the finding is. So how did they do it? Let me walk you through the experiment. you will learn a little bit of molecular biology and the powerful techniques that are now available to us to address some of these issues. So first of all, you obtain nuclei of nerve cells from the brain, as here on the left side. You collect about 50 neuronal nuclei, and you can sort those with machines, and then you can label the cells in a certain way, and you can transcribe. You can make copies of the RNA present in these isolated nerve cells with reverse transcriptase polymerase chain reaction. Now, you already know about reverse transcriptase, where you transcribe a RNA, a messenger RNA, into a DNA. Now, polymerase chain reaction was invented by the gentleman here on the lower right of the slide, Kary Mullis, a very colorful character. He won the 1993 Nobel Prize for this discovery. A very ingenious way by which you can isolate and amplify millionfold and more, if you want to, any particular sequence of DNA, for which you can develop a tag that will hybridize or bind to the sequence, and then you zip down many millionfold copies of the same sequence by providing all the bases that are required to synthesize this pearl of strings. That is the polymerase chain reaction. So that's then what folks in San Diego did. They first made a lot of copies of APP RNA that they reverse transcribed. So they had a little piece of material that would fish out and bind to the APP RNA, and then they would make multiple copies and run those on a gel to separate them by weight. And you can see here, you can probe then these different bands by hybridizing them with a radioactive original amyloid precursor protein gene fragment, and you can cut out these segments here, which are your products. This is what you have amplified from pre-existing messenger RNA in the individual nuclei of these Alzheimer's patients. And sure enough, you can identify variants. So these transcripts are not all the same. They have different breaking points and different lengths, and you can identify these variants. In the next slide, you can then see what happened. The third step is to separate different cDNA. These are the fragments that you can synthesize from the RNA, and you can see that here in Alzheimer's disease patients you find extra bands that are not seen in the normal control people. So there are DNAs, the cDNAs hybridized that do not occur in normal controls, and that is the story. Now, how about the insertion into the genome itself? And let's go to step 4. So here again is the amyloid precursor protein genes. We talked about the 17 or 18 introns and exons, and these are the different variants here in E that they identified with the reverse transcriptase and polymerase chain reaction assay. So now they cloned and sequenced these bands and found variants that had lost certain exons and had different splice junctions compared to normal controls. And here they cloned and sequenced these bands and identified a particular character here that seemed to occur frequently in the Alzheimer's affected neurons. And now you can go ahead and show whether in fact this is present in the genome itself. So you prepare genome DNA by PCR, making lots of copies, and then hybridize or interrogate these DNAs with the probes that you have constructed from your reverse transcribed messenger RNAs that we discussed earlier. And you can find that, in fact, there are these unusual fragments present in the DNA, in the nuclear DNA of the Alzheimer's patients. So they detected these so-called cDNA or complementary DNA in the genome of neurons. Complementary because it's complementary to the RNA sequence. So the RNA is translated back into DNA. This is called cDNA. And these cDNA sequences were identified in nerve cells in the genome of Alzheimer's patients. That should not occur because the gene is interrupted by multiple exons, so you shouldn't have linear cDNA sequences in the genome. However, here they were. The next slide is another way of showing this. So here the Alzheimer's patients, SAD3 here, and normal controls were compared. You can see all these extra copies and extra bands. So when you run the controls, nothing is seen, but here is the situation. So you can take genomic DNA products and use the primer set for the little sticky notes that you need to start the transcription, either from the APP gene or the precursor gene, which is the gene that produces the cleavage of the fragments. And you can, so the difference is present here in the APP gene. Now you can also use a probe to see whether these unusual APP cDNAs are present in the cell and where in the cell. It's called in situ hybridization. You can see here the punctate lesions here. This is where APP gene cDNA junctions have been located. This particular junction that is frequently found in the genome of Alzheimer's patients, in the neurons of Alzheimer's patients. So they are now detected by in situ hybridization, kind of proving the point that these genes are sitting in the cell, in the nucleus, and correspond to these unusual and very strange sequences. Okay, now you want to show also whether or not there is a time dependency. You know, Alzheimer's takes time to develop. So how does this relate to time? Here's an experiment where the unusual gene fragment from Alzheimer's patient was inserted into a cell line, into a CHO cell line, and you then stimulate the cell. You introduce breaks of DNA by exposure to hydrogen peroxide, H2O2 here, and you can open the DNA, which is repaired again and again. And this, of course, is an opportunity for unusual sequences to be inserted into that cell line. And you can see here, sure enough, if you do this, you get these extra bands in the transfected cells. However, if you have a reverse transcriptase inhibitor present, like in here, nothing happens, indicating that a reverse transcriptase-like activity is required to translate the RNA produced by the abnormal gene back into cDNA, which is then inserted into the genome. And here is the proof in the pudding of the time dependency. This is step 5 of the experiment. You wait a number of days, in this case up to 829 days, and you look at the occurrence of these unusual genetic DNA sequences in these transfected cells. And sure enough, you can see that there is a certain time dependency. As time goes on, more and more of these unusual transcripts have been reinserted into the genome. So that is the sequence, broadly speaking, of the experiments that Dr. Chung and his colleagues performed, showing that some cells in a mosaic fashion insert unusual cDNAs transcribed from amyloid precursor protein into the cell. Now, why would that be relevant? Well, there is the notion that genes apparently are much more fluid than we had previously thought. For example, it's now known that when you have neuronal activity going on in nerve cells, firing of nerve cells, there is a frequent breaking of DNA strands, as indicated in this slide. You can see here that as the strand is broken by neuronal activity, really, you open up sites where enzymes can bind to unwind and unfold the genome to allow for transcription of certain genes by RNA polymerase. So the formation of activity-induced DNA double-strand breaks, especially within promoter region, is required for an early response to a nerve stimulation. So now it's thought that something like this may be going on in the Alzheimer's nerve cells that Dr. Chung worked with, that in an activity-dependent way, as yet to be elucidated, these unusual and excessive copies of amyloid precursor proteins are introduced. So then, in summary, the amyloid hypothesis, or the theory that accumulation of the protein called beta amyloid is the cause of Alzheimer's disease, has driven research. However, treatments to target beta amyloid have notoriously failed in clinical trials. So this paper then may be part of the solution to the mystery. The thousands of APP gene variations in Alzheimer's disease provide a possible explanation for the failure of the more than 400 clinical trials targeting single forms of beta amyloid or involved enzymes. APP gene recombination in Alzheimer's disease may be producing many other genotoxic changes, as well as disease-related proteins that were therapeutically missed in prior clinical trials. In other words, the situation may be much more complicated than we had anticipated with our simplified amyloid model of Alzheimer's disease. So the function of the APP and beta amyloid that are central to the amyloid hypothesis can now be re-evaluated in light of the gene recombination discovery. So this is not the end of the story, this is the beginning of a new story of amyloid and Alzheimer's disease, and a broader story of copy number variations and the fluidity of the genome of nerve cells in response to their activity and environmental stimulation. Thank you for your attention, and hopefully you appreciated this, and we'll see you soon again for a talk here by Behavioral Health 2000.